Hi again. So we are going to finish up this chapter strong with proteins. So proteins are what make up everything. Proteins make up the hair. So if you want to touch your hair, you can say, okay, I'm touching protein. That's keratin. Our skin is made up of protein also, keratin. Keratinocytes make up our skin. Our organs are made of protein. Blood is composed of protein. Protein makes the pigment of your skin, the pigment in your eyes, pigment of your hair. Protein makes up pretty much everything you see. And all of the activities that your body does is using proteins. So what are proteins? Proteins are the polymers are a polymer, and they're made up of the monomers called amino acids. So here are the different this is what an amino acid looks like. That's not the different amino acids. There are 20 different amino acids that make up every single protein in your body. And what's really cool is the same amino acids that make up us, humans, make up dogs, cats, plants, bacteria. So these 20 amino acids make up everything. So cool. All proteins have the same structure, or all amino acids, I should say, have the same structure. They don't, all proteins don't. They have this amine group. They have a carboxylic group over here. They have a central carbon ring and a hydrogen off of it. And then they have this R group. This R group is what determines the polarity of the amino acids. That's the only thing that changes on every single. So there's 20 different amino acids, and they each have their own type of R group. And there's a picture of the amino acid, a bigger picture. All right, so proteins are made up of amino acids, and amino acids bond together through dehydration synthesis, forming peptide bonds. So when two amino acids come together, they form a dipeptide. Polypeptides have multiple amino acids, and proteins are made up of large polypeptides. So you have to have at least 50 or more amino acids to form a protein. They're formed through dehydration synthesis, forming what we call a peptide bond. And so they can be broken down through hydrolysis. There are 4 basic protein folding or protein structures. The primary structure of a protein is the polypeptide that is formed when you are making a protein. And so that's just the sequence of amino acids, and the type or the sequence of the amino acids will determine how it's going to fold up. The secondary structure of a protein then is going to either be an alpha helix or a beta pleated sheet. And these fold because the R groups have different charges. So you might have a positively charged R group, and next to that, a negatively charged R group that are attracted to each other, forming a hydrogen bond. We have other interactions between the different R groups that are going to cause the protein to fold in a certain way. The tertiary form of a protein is the actual functional form of a protein. So once a protein folds into its tertiary form, it now can have some certain function, and the structure of the protein determines the function. Remember that from chapter 1 when we were talking about structure determines function? Well, in proteins, structure determines function. So the tertiary form is the functional form of a protein, and it's going to be stabilized through covalent and ionic bonds. Tertiary form can be globular, or it can be fibrous. Fibrous is more structural, and it's going to form things like your hair, your skin. Globular is going to be moving, free moving. So like enzymes are globular proteins. A quaternary structure then is a protein that has more than one polypeptide that comes together and folds into its tertiary structure. So in the most common quaternary protein is hemoglobin, which has 2 alpha chains and 2 beta chains, so a total of 4 polypeptides come together and they fold together into their functional form. That forms a quaternary protein. Some proteins have prosthetic groups associated with them. A prosthetic group is a group that is not, that contains a non-amino portion to it. So an example of a prosthetic group in hemoglobin, the heme moiety, that's a non-hemoglobin or non-amino acid group that is found within the hemoglobin molecule and makes the hemoglobin molecule function properly. It's that heme group that actually allows for the binding of oxygen. So the structure of the protein is going to determine how the protein works. Enzymes, muscle contractions, membranes opening or closing is all associated with the structure. If we change that structure, we can destroy its function. So think about an egg. An egg has a clear viscous fluid, very thick, gelatinous, almost feeling fluid, right? And then it has the yolk, the yellow portion. When we take and we crack an egg and put it over onto a frying pan over flame, and that frying pan is hot, the gelatinous, kind of gooey, clear substance turns white. We have changed the structure. So that gelatinous, clear, viscous material is albumin. It's a protein. And we changed its structure, and when we did that, it no longer functions the same way. And that is called denaturation. So we changed the conformation, and we destroy the function. So denaturation changes conformation, and it's no longer able to revert back to its original conformation. Extreme heat and pH can change the conformation completely through denaturation, and we destroy its function. So proteins have a lot of different functions. We have structural proteins. I talked about keratin already. Collagen is another structural protein that we'll talk about. Some proteins function in communication. So I mentioned protein hormones when I was talking about cyclic AMP. Some are communicators like hormones, protein hormones, that interact with other cells. Some protein hormones, not protein hormones. Some proteins act as transporters. So you might have a channel protein inside of the plasma membrane that allows specific molecules to move from one side to the other side of a protein. Some proteins act to increase reaction rates. Those are enzymes. Enzymes catalyze reactions, and they decrease the amount of energy it takes for a reaction to occur. Some proteins act as recognition cells or recognition molecules or as protector molecules. Examples, we have tags on all of our cells that tell our other cells that we are self cells. Those are immune recognition proteins. We have antibodies that run around our body. They're proteins, and they basically bind to non-self antigens. We have proteins that function in movement. So we have motor proteins that are found in our muscles. They're also found in the cells, of our in our body cells, and they move things around within the cells. And then we have adhesion proteins that help to bind structures together. So think of those proteoglycans. So we're going to focus on enzymes because enzymes are really important. Enzymes are proteins that increase how fast a reaction occurs by decreasing the amount of energy it takes for that reaction to occur. So they're called catalysts, biological catalysts. Enzymes work on substrates. A substrate is just whatever an enzyme can bind to. Typically, enzymes are named for the substrate that they work on. And their job is they lower the activation energy needed for that enzyme or for that reaction to occur. All right, so let's look at what enzymes are doing. Here we have a reaction that has no enzyme, so no catalyst. Here we have a reaction that has one. And the reaction without the enzyme, it takes a lot of energy, which means it takes a lot of time for the reaction to occur. You have to first build up the energy to get the reaction to occur, and then you'll produce the product. So most reactions, all reactions will eventually occur because the molecules are constantly moving, but the amount of time it would take for the reaction to occur would be so slow that it would not allow for life to exist. Here with the enzyme, the activation energy is super small, so we can produce the response very rapidly. And so enzymes work on their substrates, and they do this by having the substrate bind to a region called the active site. The active site is a region where the substrate can bind, and typically enzymes and substrate form a bond that we used to call a lock and key fit, where the enzyme binds and it fits perfectly. But now we know that it actually is more like an induced fit. The substrate binds and the enzyme kind of curls around or gives the substrate a hug to form a response. So here we have sucrase as the enzyme, sucrose is the substrate. It binds into the active site, and the sucrase will catalyze a reaction when they bind and kind of give a little hug and produce the product, glucose and fructose. This is just a bigger picture, not a bigger picture, a full picture of what you saw before. Oh, why are you not working for me? Okay. If you are interested in seeing this video, you can look up enzyme action and the hydrolysis of sucrose in the in your Google tab, and it'll pop this up, okay? So enzymes, when they work, they do not get used up. Enzymes can work again and again and again without being consumed. And this is really important because this means we don't have to produce multiple enzymes. We can use the same enzymes over and over again so long as we don't damage them or denature them. And they can catalyze millions of reactions every single minute. So you can have one sucrose that breaks down one sucrase that breaks down tons or millions of sucrose molecules every single minute. Which means if you have a lot of sugar in your system, you're breaking it down really fast. There are different factors that will affect enzyme reaction, though. If we increase the temperature too much, then that's going to affect enzyme activation. So if it gets too high, it will actually cause enzymes to stop functioning. Slight increase in body temperature can actually make enzymes work a little bit more effectively, which is why a fever can be beneficial. Changing the pH, either increasing or decreasing the pH, can actually affect enzyme activity. So enzymes that work in like a pH of 7 will not work as effectively when the pH drops. So salivary amylase, which works really well in your mouth, where you have a neutral pH, will not work when it enters the stomach. But pepsin, which works on proteins in the stomach, will work really well in the stomach, but as soon as it moves into the small intestines where we have neutralized the environment, will not work anymore. Some enzymes need helpers or cofactors to work effectively. A cofactor is an inorganic molecule that works with the enzyme. A coenzyme is an organic molecule that works with the enzyme. And I typically think of an enzyme is something that catalyzes a reaction, and then the coenzyme just helps that reaction work more effectively. And that's not going to work either, so I'm not going to worry about playing this. Actually, you know what? Hold on a second. So this is the animation. I wanted you to be able to see it, so here we go. Or
this animation
there we go.
Demonstrates how B vitamin coenzymes must be present in order to enable some enzymes to carry out their functions. Many of the B vitamins assist in the complex biochemical processes that produce energy for food. The B vitamins are essential parts of coenzymes. Coenzymes are part of the machinery that allows reactions to take place. Enzymes are large protein molecules that catalyze biochemical reactions by lowering activation energies. Enzymes accomplish catalysis by configuring chemical reactants in the best possible spatial arrangement for reactivity. This enzyme catalyzes the release of carbon dioxide when producing energy from carbohydrates. Combining with the coenzyme helps the inactive enzyme achieve the appropriate three-dimensional configuration. For this reaction, thiamine must be present as the coenzyme. The green coenzyme molecule above is thiamine pyrophosphate, the cofactor needed to permit the enzyme and the substrate, pyruvate, to combine in the correct orientation to catalyze the reaction. Now all three molecules are nested together, and the substrate, pyruvate, combines chemically with the coenzyme, thiamine. The biochemical reaction can take place, and energy as ATP and CO2 are produced as products of the reaction. Once this happens, the remaining complex, containing the two carbon fragments, thiamine and the apoenzyme, rearranges and releases the two carbon fragment, acetaldehyde and ATP. The departure of the acetaldehyde opens the thiamine pyrophosphate to combine with another molecule of pyruvate, and the cycle continues. Meanwhile, the energy is now in the molecule ATP and is available to convert or to be stored. Another way that a coenzyme can influence the reaction between substrate and enzyme is to cause a change in configuration of the enzyme without interacting with the substrate. Here you see the conformational change in the enzyme needed to configure the enzyme to accept the substrate in a lock and key complex. This is called allosteric control. The cofactor approaches the inactive enzyme and binds to it in an enzyme-coenzyme complex. The new configuration is a good fit for the substrate. Once this enzyme cofactor substrate complex is formed, energy can be released as ATP, and other reaction products can be released from the complex. Now the correctly configured active site on the enzyme is free to engage with other substrate. The amount of B vitamins that are needed to serve as coenzymes is quite small. There is enough vitamin activity in 1.2 milligrams to meet the average person's needs all day. Extra B vitamins will not produce more energy. excess B vitamins in the body will pass out of the kidneys into the urine. This table shows some details about several of the roles played by B vitamins as enzymes.
All right, I'm going to stop this and we'll go back to our lecture. All right, and okay. So let's talk about, or we talked about enzymes and how they work. But in general, we don't just have one substrate that has to produce or has to go through a metabolic reaction to produce some type of a product. In most cells, we have pathways that are followed. And so these pathways are called metabolic pathways, where you have multiple enzymes producing products, and those products become the reactants in the next reaction. And most metabolic pathways are very complex. They're not just straight 1 to 2 to 3 to 4. They can branch off. They're very branching. So I'm going to explain it to you in a very simplistic form, but just please understand that this is not simple at all. So, darn it, sorry. Oh, man, okay. A metabolic pathway is a pathway where you have one reactant that is going to be transformed by some enzyme to a product. That product becomes the reactant in the next metabolic activity, or the next, I should say, metabolic step. So here we have A is transformed by alpha enzyme to B. B is transformed by beta enzyme to C. C is transformed by gamma enzyme to D. So A is the initial reactant, and the end product, overall end product, is D. B and C are intermediates of this pathway. And so metabolic pathways can be controlled, turned on and off, depending on if we need D or not. Make sense? This is another another video that you can watch. It's not going to play on here. I'm not as concerned. I think the idea of an enzyme converting something from one to another is not that hard to understand. So I'll let you watch this one on your own. Again, all you have to do is type in McGraw-Hill, a biochemical pathway, and it should pop directly up in your Google bar, okay? So, question for you that you can think about. I'm not going to give you the answer, but Mrs. Roberts is in a diabetic coma. She's just been admitted to the hospital. Her blood pH indicates that she is in severe acidosis, and many measures are quickly instituted to bring her blood pH back to normal. So, why is acidosis a problem? And while you're answering that, tell me what the pH is and what normal pH of blood is. Oh, I'm not going to give you the answer, but the answer is on the next slide, so I'll stop right here. Okay? Actually, I'm going to flip over. Okay, yep, I am stopping right here. Have a good day, and I will see you on Wednesday. And